In this class, let's put necrosis and apoptosis side by side and understand the differences. I'm Nabeel. This is Pathocyte. Let's start with the cause. Necrosis is generally caused by severe pathological injury. For example, severe ischemia, infections, toxins, burns, and other physical or chemical injuries can cause necrosis. So, necrosis is usually an unwanted consequence of severe cell injury. Apoptosis is different. Apoptosis can occur as a normal physiological process where the body deliberately removes cells that are no longer required. For example, during development, say during the formation of fingers, the excess tissue in the form of web is removed by apoptosis. During normal cell turnover and during the elimination of certain immune cells. But apoptosis can also occur in pathological conditions. For example, a cell with severe DNA damage or excessive accumulation of misfolded proteins may undergo apoptosis. So, apoptosis can be physiological or pathological, whereas necrosis is usually always pathological. Necrosis generally affects large groups of cells. For example, if a blood vessel supplying part of the heart is blocked, many myocardial cells in that area can undergo necrosis. An infection can also produce necrosis involving a relatively large area of the tissue. Apoptosis, on the other hand, usually affects individual cells or small groups of cells. For example, during normal cell turnover, one cell may reach the end of its usual life and undergo apoptosis, while the surrounding cells remain completely normal. This allows the body to remove individual cells without causing widespread damage to the surrounding tissue. Now let's look at the size of the cell. In necrosis, the injured cell typically swells. Severe injury interferes with the cell's ability to maintain its normal ion and water balance. So, water enters the cell and the cell becomes enlarged. In apoptosis, the opposite happens. The cell shrinks. The cytoplasm becomes dense and eosinophilic, and the cell eventually breaks up into small apoptotic bodies. So, necrosis is associated with cell swelling, while apoptosis is associated with cell shrinkage. And this brings us to one of the most important differences, the plasma membrane. In necrosis, the plasma membrane becomes progressively damaged and eventually loses its integrity. Once the membrane is disrupted, the contents of the cell can leak into the surrounding tissue. In apoptosis, the plasma membrane remains intact, but the membrane undergoes changes and the cell eventually breaks up into membrane-bound apoptotic bodies. So, even though the cell is dead, its contents remain packaged inside membranes. Now, think about what happens to the contents of the cell. In necrosis, because the cell membrane is damaged, cellular contents can leak out. At the same time, lysosomal enzymes can contribute to enzymatic digestion of a dead cell. In apoptosis, the cellular contents don't simply spill out. The cell is fragmented into small membrane-bound apoptotic bodies. These contain portions of the cytoplasm, organelles, and sometimes nuclear fragments. They can be rapidly engulfed by phagocytes. This brings us to one of the most clinically important differences: inflammation. Why does necrosis usually produce inflammation? Because the membrane is damaged and intracellular contents are released into the surrounding tissue. These released molecules act as signals of cell injury and can trigger an inflammatory response. So, inflammation is a common feature of necrosis. In apoptosis, the situation is very different. The cell remains enclosed within membranes and is broken into apoptotic bodies. These fragments are rapidly recognized and engulfed by phagocytes. Because there is almost no leakage of intracellular contents into the surrounding tissue, apoptosis usually does not produce a significant inflammatory reaction. Another difference is what happens to lysosomes. In necrosis, severe injury can damage lysosomal membranes. Lysosomal enzymes are then released into the cytoplasm and contribute to the enzymatic digestion of the injured cell. In apoptosis, the cell is dismantled through its regulated intracellular machinery, basically the caspases. The lysosomes themselves are not responsible for producing these changes in apoptosis. Now let's come to the nucleus. This is an area where the two processes have some overlap. Both necrosis and apoptosis can show chromatin condensation and nuclear fragmentation. So, pycnosis and karyorrhexis are not exclusive to necrosis. In necrosis, the classical sequence of nuclear changes is pycnosis, karyorrhexis, and karyolysis. Pycnosis means shrinkage of the nucleus with condensation of the chromatin. Karyorrhexis means fragmentation of the pycnotic nucleus. And karyolysis means fading and dissolution of the nucleus, largely because of degradation of DNA. In apoptosis also, chromatin becomes condensed, but here the condensation is under the nuclear membrane, basically peripheral part of the nucleus. The nucleus may then fragment as the cell is being dismantled. But unlike the progressive nuclear destruction which happens in necrosis, here in apoptosis, this is part of a regulated process of cellular fragmentation. There is another important difference in what happens to the DNA. During apoptosis, executioner caspases activate enzymes like DNase that degrade the cell's DNA. The DNA is cleaved into fragments corresponding to multiples of nucleosomes. Because this fragmentation is relatively organized, when DNA from apoptotic cells is separated by electrophoresis, it produces the characteristic DNA laddering pattern. In necrosis, DNA degradation is much less organized. So, electrophoresis produces a more diffuse smear rather than a ladder. This is a nice example of how molecular mechanism produces the laboratory finding. Finally, what happens to the dead cells? Both necrotic and apoptotic cells eventually have to be removed. But the context is very different. In apoptosis, the cell is broken into small apoptotic bodies, which are rapidly recognized and engulfed by phagocytes. One important signal is the exposure of phosphatidylserine on the outer surface of the apoptotic cell membrane. This acts as an eat me signal for phagocytes. So, apoptotic cells can be removed very efficiently, often before their contents have a chance to leak out. Necrotic cells, on the other hand, are removed in the setting of tissue injury and inflammation. The cellular debris is eventually cleared by phagocytic cells only. There is also an important clinical consequence of the difference in membrane integrity. When necrotic cells lose their membrane integrity, intracellular proteins can leak into the circulation. We can use these proteins as markers of tissue injury. For example, when myocardial cells undergo necrosis in myocardial infarction, cardiac troponin is released into the blood. Similarly, enzymes such as transaminases can increase in blood when hepatocytes are injured. So, the leakage of intracellular contents that makes necrosis inflammatory can also give us useful biomarkers of tissue injury. So, let's put everything together. Necrosis is always pathological, while apoptosis can be physiological or pathological. Necrosis affects a large group of cells, whereas apoptosis can occur to a single cell or a small group of cells. When it comes to cell size, a necrotic cell swells up, whereas an apoptotic cell shrinks. The plasma membrane is disrupted in necrosis, but in apoptosis it is intact but altered. Cellular contents may be digested and may leak out in necrosis, whereas in apoptosis they are packed into apoptotic bodies and remain intact. Inflammation is frequent in necrosis, whereas it is usually absent in apoptosis. The membranes of the lysosomes are affected in necrosis and the lysosomal enzymes can leak out, whereas in apoptosis, lysosomes more or less remain intact. The nucleus undergoes pycnosis, karyorhexis and karyolysis in necrosis, whereas in apoptosis there is chromatin condensation under the nuclear membrane and later on nuclear fragmentation which occurs. DNA electrophoresis shows smear-like pattern because of irregular degradation in necrosis, while in apoptosis a DNA laddering pattern is seen. The dead cell is cleared by phagocytosis after tissue injury and inflammation in necrosis, while the apoptotic bodies are rapidly phagocytosed in apoptosis. So, necrosis and apoptosis both result in cell death, but they represent two very different ways of getting there. And that brings us to the end of our discussion on cell death, from necrosis to apoptosis. And if you've been following this series from the beginning, I hope the whole picture is starting to come together. If you are finding these classes useful, consider subscribing to Pathosite. I'll see you in the next class. Think at the cell level.